in mind, we can move back to wrapping up our discussion on metabolism and then moving into photosynthesis. I think I left off here. I got through this slide, right? And that's where I finished? Okay, so this was an example of an experiment where they're asking the question, does this thing really spin around? At the time, it was very controversial. The concept of chemoosmosis, like using a potential energy gradient to generate ATP in mitochondria, and a turbine, they were both very controversial. Now, there's loads of experimental evidence to support it, but this was some, and I discussed this in the last lecture, with some of the earliest sort of data. And again, it draws back on our discussion about confocal microscopy, fluorescence microscopy. Could be confocal, wouldn't have to be. Just using a fluorescent tool and a fluorescent sensing microscope to answer a fundamental molecular question. Okay, so now we have this big picture of these metabolic pathways. And the most important concepts, of course, are what are they for? One of the answers was always, of course, energy transformation. But another answer to that, not with the ETC, but certainly with glycolysis and Krebs, other pathways we haven't talked about, is providing carbon skeletons to do other reactions in the cell. We'll look at that in a minute. Where do they take place? How are they regulated, etc.? So we looked at glycolysis feeding in with the sugar at the, quote, top of it. All right? And we said, well, you get down to CO2 and water, lots of energy is available. But this is a nice diagram in the sense that it shows you the metabolic flexibility of some of these primary pathways in nice, simple terms without getting bogged down in the structures and many names or enzymes. If you look at carbohydrates, you can feed them in, sure, of course, 6 carbon sugars at the top of glycolysis, or you can feed carbon skeletons further down in glycolysis. The further down you go, if you start to bypass and feed in a carbon skeleton quite low, you won't get as much energy yield out of it. If you're feeding in at the bottom, say with a pyruvate carbon skeleton. But you can still feed into it. So for example, proteins. If carbohydrates and fats aren't available, proteins are still partially oxidized. There's still quite a bit of energy in a protein. You can metabolize protein through these pathways and get energy out of it. You have to deaminate it. That's the ammonia coming off of the side there. You're not going to get electrons off ammonia. But you can oxidize the carbon skeletons of amino acids. So chop up proteins into amino acids, chop off any amine groups or nitrogens, and then feed that partially oxidized carbon skeleton into primary metabolism, either at the point of pyruvate or 2 carbons into acetyl coenzyme A, or depending on what the amino acid was, the carbon skeleton will go directly, it could go directly into the Krebs cycle or be metabolized a bit first so that it's a metabolite of the Krebs cycle and feed it in. So there's metabolic flexibility. Fats, we didn't talk about fat metabolism. I've chosen just a couple of the main primary pathways. But I have emphasized, because carbon is so much more reduced in a fat molecule than in something like a sugar where you have oxygens, it's partially oxidized, it's a longer distance in terms of oxidation for fats to CO2 and water than it is for a sugar. So you get more energy. And when you feed the fats, the fatty acids, you can take 2 carbons at a time off them, link them up to coenzyme A, and now they're perfect substrates for going into the Krebs cycle. But you can also, with the triglyceride, once you've taken those fatty acids off and you're left with that glycerol backbone, you can feed that into glycolysis as well. Right? So this is the flip side of the coin of these pathways. You can siphon carbon off and use it to synthesize important molecules in the cell. You need to. But you can also feed into them. Right? So that metabolic flexibility. Okay. It's an important concept to keep in mind. But if we're getting energy out of glucose, or any of these carbon skeletons that we're feeding into these pathways, if we're taking it all the way down to CO2 and water, well, if you're taking it down to water, that means you're reducing oxygen. That's the big bang for the buck. If you stop just after glycolysis, you get very little energy out. You're left with pyruvate, and there's still lots of energy in pyruvate. So what happens when oxygen isn't available? For us, it's not a good situation. We can't go very long without oxygen. Other organisms can substitute other electron acceptors. nitrates, sulfates, irons, all kinds of things. But we can't do that. So we need oxygen. If oxygen isn't available to take the electrons in the electron transport chain from NADH, FADH2, or other donors, those are the most common, then that chain stops. The gradient's diminished. You're not making ATP there. NADH, FADH back up. What's the consequence of that? Well, you should know. Those are high-energy compounds. They're going to back up. They're going to inhibit the Krebs cycle. So the Krebs cycle shuts down when there's no oxygen. So even though the Krebs cycle, you look at it and you go, oh, it doesn't require oxygen. It does, because it's so tightly associated with the activity of the ETC. So it shuts down. And you're left with just glycolysis. You're left with a low payoff. I'm never going to ask you to churn these numbers out memorization style. Just get a feel for the ratio. It's almost 10 times difference. It's a big difference, because those values will actually change depending on the cell condition. So you're stuck with glycolysis if you don't have oxygen, and glycolysis is going to accelerate. It's going to go faster because it's trying to keep up with the ATP demands of the cell. It's called the Pasteur effect. But in order to keep glycolysis going, pyruvate can't be the end product, because one of the things glycolysis needs is NAD. And if you're not getting it from the electron transport chain, NADH donates its electrons, so now it gets oxidized. NADH gets oxidized, it's NAD. NAD can go back to glycolysis and the Krebs and keep them going. But if you're not using up the NADH in the electron transport chain, you can't keep glycolysis going without feeding NAD into it somewhere. It's required in glycolysis. So different organisms approach that problem in different ways. We produce lactate, not lactic acid. So I mentioned this is one of the most common biochemical misconceptions in biology. Very well-established, important textbooks even still show this error. You can't make lactic acid. You make the opposite of it. You make the base from a base. Pyruvate gets converted to lactate. In fact, you look at that reaction, it's consuming a proton. Right? They're showing 2 here, so 2-pyruvate, 2, etc. So each pyruvate conversion to lactate pulls a proton out of solution. It's resisting acidosis. This is an adaptation to acidosis, not a contribution. It's the opposite. You're synthesizing a base from a base. And this is a cytosolic pathway. The pH of the cytosol is around neutral. All of these organic acids, lactate, succinate, pyruvate, alpha-ketoglutarate, whatever it is, are present in the form of bases. Does pH go down under anaerobic conditions when lactate's being synthesized? Lactate goes up, pH goes down. This is not causing this. That reduction, that acidosis, is caused by other reactions that are also occurring under anaerobic conditions or hypoxic conditions. So this guy who's getting jacked at the gym, he thinks I'm getting lactic acid burn. You're not. You don't make lactic acid. You make lactate. It's something of a dead-end product. In us, it isn't. There's still lots of energy in lactate, and it will get metabolized in other tissues, typically from muscle. If we're talking about muscle, it'll get moved and ultimately brought back into primary metabolism so you don't lose that carbon. But there's still lots of energy in lactate. This isn't really about getting energy. In fact, it's consuming NADH, a high-energy product, because it needs the NAD to keep glycolysis going. That's the point of that reaction. And lactate production is also not the cause of muscle ache after, say, an anaerobic workout. That's muscle damage. It's caused by other things. Or the pain, the discomfort is caused by other things. Other organisms don't make lactate, but they have the same principle. Regenerate the NAD to keep glycolysis going, because that's all you got going for the lack of oxygen. So glycolysis is a very ancient pathway. It predates the high oxygen levels on the planet. And so it can run in the absence of oxygen. But it needs a source of carbon coming into it. You can see there's a few different carbon skeletons you can feed into it, but works best when you're feeding at the top with 6 carbon sugars. It needs that NADH to keep going. And of course, ADP, because you're going to make ATP. Some of that ATP is going to be used to keep glycolysis going as well. You use up ATP, you generate ADP, you can keep using it in glycolysis. Okay, If you're producing ethanol, so pyruvate, oxidation acetaldehyde, and then ultimately to ethanol, conversion to ethanol by ADH. This is an end product, a waste product. Even though you know there's lots of energy in ethanol, put a match to it, you see that instantly. But microorganisms like yeast, let it diffuse away. Plants, this is their approach to low oxygen, produce ethanol, let it diffuse away. It's a waste, losing energy, but that's okay. It keeps glycolysis going. We benefit from it. Obviously, we enjoy the production, the metabolism of yeast that can take glucose and ultimately, under low oxygen conditions, convert it to pyruvate, ultimately to ethanol, and CO2, so you get a carbonated beverage with alcohol. You may be familiar with some of them in your trials, tribulations, travels. Of course, there is a side to that that's the darker side. Ethanol is a toxin, and so are the byproducts of its conversion ultimately to something metabolizable. So we evolved, probably the reason mammals evolved to detoxify ethanol is because of fermenting fruit. So fermenting fruit, ethanol gets produced. If you eat fermenting fruit, you're taking in that toxin. So mechanisms, there would have been selective pressure to evolve mechanisms to detoxify that. So this combination of enzymes, alcohol dehydrogenase, acetaldehyde dehydrogenase, convert that ethanol into acetate. And particularly, you don't want acetaldehyde kicking around. It's a very toxic compound. This is what you guys covered in the labs this week. But the genetics of the ability to do that vary among the human population. And no surprise, they vary geographically. So genetics works. So northern Scandinavia, a lot of Asian populations have either mutations or just different alleles, so different forms of the same gene, of acetaldehyde dehydrogenase, I think also ADH, and impairs their ability to metabolize this. So the acetaldehyde builds up, you see this red flush, faster hangover, more severe hangover. So just something to keep in mind. But once you have a small 2-carbon compound like acetate, no problem. You can get that back into primary metabolism. You don't want those toxins kicking around. That's my Uncle Bill here. It's not my uncle. Ethanol, interestingly, is still used as an antidote, and this was in your lab, for certain types of poisoning, methanol poisoning and ethylene glycol poisoning. So often pets or kids get into ethylene glycol washer fluid, windshield washer fluid, because it has a sweet taste to it, but it's very, very toxic. But now that you understand the principles of competitive inhibition, or outcompeting something, I think in this case, they get converted into toxic products, so they're essentially alternate substrates. But the enzymes are better with their natural substrates, ethanol. So flood the system with ethanol, and then the odds of if there are 10 ethanol molecules to every methanol, the odds of methanol getting in there, getting converted to something even nastier, are very low. And your system will ultimately flush it out. But if you're in a merge on an ethanol drip, sure, it'll save you from methanol poisoning, but you're going to get a wicked hangover out of it, that's for sure. So I think they have been developing alternatives to ethanol as an antidote. But the point I'm really making here, it sounds like just kind of an interesting anecdote, is it really reinforces these principles of how active sites work, how similar shaped, sized molecules with similar chemistry, can get into the active site, either as competitive inhibitors, alternative substrates, toxins. All right, so it's just some notes for these couple of slides here, just for your notes. I want to come back to this point that there's 2 points. So one is fairly obvious, but I really want to reemphasize it. Of course, the most important question for what's the point of glycolysis, the most important questions are what is it for? What's glycolysis for? What's Krebs cycle for? What's the ETC for? And for those metabolic pathways, Krebs and glycolysis that we've looked at here, it's energy, transformation, capture some of it, high energy molecules, that's one. Production of a good fuel for Krebs, et cetera, et cetera. But one of the answers is always to provide carbon flexible carbon building blocks, flexible in terms of utilization, not necessarily flexible structure, for other reactions in the cell. And we saw with that metabolic flexibility diagram, carbon skeletons coming in different places, but what about going out? So I'm going to wrap up this section with a couple of examples of a secondary, usually now we actually call it specialized metabolism. And this is more common, these specialized metabolites are more common in microbes. Remember, there's tens of or hundreds of thousands of species of microbes on the planet, and many plants. They're of great biological interest. They deter predators. They deter herbivores if you're a plant. They're allelopathic, meaning they poison others who are trying to compete in your little space. They draw pollinators, etc. So biologically, ecologically, these are tremendously important. Cells need the ability to synthesize them. But commercially, they're also hugely important. Medicines. Vast amount of our medicines either come directly or indirectly from plant or microbe sources, not just antibiotics. Other commercial industries, like besides the pharmaceutical and the medical industries, perfumes, spices, things we like, compounds that are healthy for us, antioxidants, etc. So it's worth visiting. So take a brief side. This one I like to show, not because it's got a shocking diagram, but because it's a warning. This stuff is around here. There's a huge patch of it right at the end of Sir John A. Macdonald, where the 401, where you'd make the curve onto the 401. It's senescing now. It looks like Queen Anne's lace. It's in the wild carrot family. It's wild parsnip. Well, giant hogweed is similar, but it's bigger. Has anybody been burned by this stuff? Almost every year. Yeah, one person. Was it this bad? No. But it depends how sensitive you are. I know I have colleagues who've been burned this bad by this stuff. It leaves wicked scars. So these plants produce a compound, furanocoumarins. They're a herbivore deterrent. You're not going to go back to that. You get damage like this. But they're activated by UV. So you walk through, and this stuff is all around this area, invasive. You walk through a patch of this stuff in the day, the UV activates it on your skin, and you get nasty chemical burn. You walk through it at night, take a shower, no problem. So something to keep in mind. But it's the carbon skeleton of all of these things I'm going to show you is drawn from primary metabolism. And there are thousands of these kinds of compounds, at least in the hundreds, if not thousands, in any given plant. Another point is that, of course, there's always an evolutionary battle between, in this example, herbivores and the plants. So one of the herbivores on this is a parsnip worm, like a caterpillar, a larva, and it's also sensitive to the toxin, but in order to avoid it, instead of having a chemical detoxification system, it uses a behavioral adaptation. It takes the leaf, wraps it up in silk, kind of curves it around, and it has a shaded compartment, and just eats it in the shade. No UV activation, no problem. So you see loads of examples of this as you continue in biology, and of course I hope you do, of these ecological adaptations and the evolutionary battle between parasites and hosts and herbivores and plants, and etc. These are, of course, compounds that are familiar to you, probably at some level, delicious but deadly. These are a group of compounds called alkaloids. They're among the most medically important compounds, valuable and efficacious, so functional, on the planet. Morphine is still a very, very difficult, you can't synthesize it artificially, or at least it's impractical. And also the structure of this, the backbone of it, again, drawn from carbon from primary metabolism, shunted away into complex reactions that give you this and all kinds of other structures. Cocaine, nicotine, caffeine, these are all anti-herbivore compounds. Nicotine is quite toxic. Capsaicin, here's an interesting one. You know it's a deterrent. You bite into a hot pepper, and you can feel it. You feel it coming and going, typically. It activates heat receptors. That's why it feels hot. It activates a heat receptor. So it makes a good anti-herbivore deterrent. But birds don't have that heat receptor, or they have a different allele, a different form of it. So they're not susceptible. Birds can eat these, at will, with suffering no ill effect. And so they're a great vector. They eat the pepper, fly around, drop the seeds in their feces, plant achieves what it wants, spreading its genes. Bird gets some food, works well. But capsaicin as a pure compound is nasty. You have to wear a full bodysuit if you're working with pure capsaicin. Okay, atropine, also you see this plant around here, deadly nightshade. It's in the potato, the Solanaceae family. It is still used to treat bradycardia, so irregular heart rhythm. I think it's still used in some cases in emergencies. Also, nerve gas antidote. But the point is, these complex carbon skeletons. There's way more complicated ones. This stuff, lignin. A picture of a lignin molecule, you can Google what a lignin molecule looks like. It's insanely complex. It's got a lot of repeating units, but it's not just pure repeating units. All of that's drawn away from primary metabolism. Terpenoids, this is a huge class, very important in medicines, very important things we like mint, so spices, fragrances. Here's one class of them that plants make to look like hormones, insect hormones, even human hormones. Some of the compounds, phytoectozones that are produced by soybean, for example, are very similar to human hormones. Here's a three-headed larva. So its developmental pattern is seriously perturbed by feeding on plant tissue that has these phytoectozones. But again, this is drawn from primary metabolism. And one last particularly cool example is a multitrophic level one. So wild tobacco, it's not really tobacco, it's related to tobacco. It's called Nicotiana. Nicotiana is in nicotine. It's a tobacco family, but the Ammiana grows in arid environments. It has a natural herbivore predator, the tobacco hornworm, great name, which is resistant to nicotine. Nicotine is a very nasty compound. Nicotine kills most insects. Doesn't do you any good either. But the nicotine hornworm has developed chemical resistance to nicotine. So the plant, again, always this evolutionary battle, the plant releases volatile compounds, volatile terpenes. So carbon skeleton came from primary metabolism. It's a defense compound. It's detected by wasps in the environment, parasitic wasps. They follow that chemical scent, high affinity. They must have high affinity receptors, just like those Drosophila you find in your when you open a bottle of wine. They follow it to the source, target the hornworm, lay eggs in it. The eggs hatch. They eat the hornworm from the inside out. And it's another example of beautiful natural interaction. But in addition, the female adult moths, they sense those volatile terpenes as well, and they avoid plants that are producing it because they know the defense is coming of the parasitic wasps. So this is a great multitrophic level example. And if I put two tobacco plants like this together and infect one with hornworms, this starts producing volatile compounds. This plant will detect those compounds and start to mount defenses, even though it's never seen a predator. It's not this plant helping this plant out. This is not akin altruistic action. Hey, buddy, I'm under attack. This is just eavesdropping. It notices, detects these compounds, and that's that information. So there's information processing that triggers defense activity. Okay, so I have to log back into this. I want to do, as a wrap-up to the metabolism stuff, one question from Can you see the question? Whoa. Okay. There we go. Okay. Sometimes this gives me a better display, sometimes it doesn't. But you should be able to see it fine. This is a trickier question than you think it is. Read it carefully. This is a previous exam question. Why can't I make this bigger? Let's just do this. Okay, I can't squeeze it all in there for some reason, but You should be able to see it on your screen. Okay, so let's see what people are thinking. You got A, B, C, D, E. It's moving around. It's moving around. I'm going to tell you that the answer is not E. I'll tell you why in a second. All right, I'm going to stop and get your bestest guestest in. Okay, so you got enough ATP for a very short period, but you need more. You're low on supply. First of all, it doesn't say anything about oxygen availability. And although we just talked about that, that's probably why people defaulted to that. All that's going to happen, remember, the cell is about regulation, adjusting metabolism to the metabolic needs, adjusting catabolism, adjusting anabolism. You need more ATP, more catabolism, less anabolism, and vice versa. So it's actually, in a sense, a simple question, but the distractor, because you know that under low oxygen, which may or may not be the case, the question doesn't say anything about oxygen availability, but the answer is simply catabolic processes are going to be activated to generate more ATP, right? And if you don't have enough glycogen, so you're not feeding enough carbohydrate in, you got lipid and protein. You need to have something. Carbohydrates, lipid, protein. That's what you're burning. Okay. So now, wrapping up metabolism, try to keep in mind the big pictures. And as we move into the next major energy processing context, try and draw parallels. How are mitochondria and chloroplasts similar structurally? How are they biochemically similar? What are the roles of, same questions. What goes in, what comes out? How is it regulated? What's it for? This is where all the energy starts on the planet. This is where most of the carbon starts on the planet. So you can't overemphasize the importance of photosynthesis. You end photosynthesis, you end the complex life on this planet. This is a structure, obviously not an actual image, but a diagram of a typical chloroplast structure. The first thing you should notice about that, and draw that structure-function correlation, is, wow, look at all that membrane surface area. That is also one of the first things you notice about a mitochondrion. Two different powerhouses with some similarities and some differences. They both use, well, let's look at some of the similarities. They both have a lot of internal membrane surface area. Why? For mitochondria, it's to accommodate lots of electron transport chain activity. That's where the electron transport chain is. Remember, you need to know where something is, and you can't just say in the cell, cytoplasm, mitochondria. ETC, inner mitochondrial membrane. You want lots of ETC activity because that's where bang for the buck is. You want lots of membrane surface area. Chloroplast, these are your solar panels. That's where chlorophyll is on those thylakoid discs, on the membranes. You want lots of solar panel to capture lots of energy. That's why there's so much. Right? Structure-function relationship. Another similarity is the use of potential energy gradients, so electrochemical gradients, for ATP synthesis through that turbine ATP synthase. We'll look at that in more detail. So this just moves up kind of in hierarchy, or down, if however you'll prefer to look at it, from the leaf level anatomy down to the ultrastructure, emphasizing all of that interior membrane surface area. And in between, the solution surrounding those membranes, so they're called thylakoid discs, thylakoid membrane, that's where the light-absorbing pigments are. The solution that surrounds them is analogous, metabolically is what I mean, to the mitochondrial matrix in the chloroplast called the stroma. right? So the matrix and stroma have metabolic similarities. The membranes of chloroplast and mitochondria have metabolic similarities. So you should be comfortable kind of drawing those parallels. This is another one of these simple summary diagrams that I like very much. If you can take a diagram like this and explain it, not just redraw it, but actually explain it to someone in perhaps a couple of different ways, then you understand photosynthesis, because it's showing you both the light capture reactions and the carbon fixation reactions. This is where there's two of the most important processes, you could argue the two most important processes biologically on the entire planet. The capture of light energy and the use of that light energy as it's transformed to other types of energy to capture carbon and make building blocks. Those are the two most important processes on the planet. This is where carbon enters the biological world. It goes from an inorganic form, CO2, no energy in it, to ultimately a higher energy form, and not just sugar. It's the building block for everything, and not just in plants, everything that eats plants, right? I know that you already know this, but I think it's definitely worth emphasizing how important that is. And again, it's about electron transfer. The point of glycolysis, Krebs electron transfer chain, was to get electrons from glucose, or whatever your reduced carbon form is, oxidize it, take electrons off it, loss of electrons oxidation, and move them through a series of carriers to lower and lower energy levels until you put them on oxygen and you end up with water. And as you oxidize CO2, it goes from, it's reduced to valuable forms to highly oxidized CO2, not so valuable, except for the photosynthetic world. So when you excite an electron, you can either use it to do chemistry like we looked at, or it can return to its unexcited state and just release that energy, say as in the form of light, fluorescence, light, or heat. But plants use that energy of excited electrons to do work. They move electrons around, just like in the pathways we looked at, to lower and lower energy electron carriers, capturing some of that energy in the form of other potential energy, gradients and chemical bonds. But who does that energy capture? Well, you know this. It's chlorophyll. But it's not just chlorophyll, it's other pigments. And again, based on the lectures we've had so far, you should be able to look at molecules like that and infer things about them, especially the carotenoids. There isn't a polar bond there to be seen. That is a very insoluble molecule. It doesn't like water, it's hydrophobic. So you want it in a position to be able to capture light. It needs to be in a hydrophobic environment. So, because it's hydrophobic likes hydrophobic, the membrane is a perfect, thylakoid membranes are a perfect environment. Same with chlorophyll. Very long hydrophobic tail, can be poorly soluble, but will integrate very well. It's a very nonpolar molecule. It will interact very well in a lipid environment. Hence, the thylakoid discs. But how do we know these are the pigments that are responsible for photosynthesis? That's what the point of these images are. Every time you see a diagram in your notes or the textbook if we're using the diagram from the textbook, don't ask yourself, can I redraw this? Just ask yourself, what does this mean? What is this diagram trying to tell me? This is showing you the wavelengths that these pigments absorb at. This panel is showing you the wavelengths of photosynthetic activity. If you take a leaf and give it different wavelengths, what wavelengths give you oxygen production? Oxygen's a byproduct. It's not what the plant is after. Maybe the mitochondria capture and use it, great. But you can measure oxygen production as a proxy, a measurement of photosynthetic activity. It's in the blue and red spectrum. It's not in the green. You know that. Plants reflect green light. That's why they look green. Now, if you compare that action spectrum to the absorption spectrum, it matches these particular pigments. There's lots of other pigments in plants, kinds of anthocyanins, all kinds of things. But they don't match that action spectrum. So we know, because of experiments like this, that these are the pigments responsible. And certainly since that time, lots of other experiments have been done to support that. It's fact. Right? So that action spectrum matches the absorption spectrum. We know that those are the the molecules that are responsible for light capture. And again, you can't overstate how critical light capture is and conversion for life on the planet. So how does that work? Again, a nice simple diagram. I've always, for this kind of stuff, I've tried to find the simplest diagrams I could, not the really complex ones with lots of names of the electron carriers. Think of it in the bigger picture. Light is captured. You just saw that. It's absorbed. Light is captured by those, the energy of light is absorbed, if you will, by those pigment molecules. That excites them. So electrons get excited. They can transfer that energy to a specialized chlorophyll in photosystem 1 and photosystem 2. These specialized chlorophylls will do the chemistry. So the energy gets transferred from one pigment molecule to the next in a downward way. That is to say that it's like a funnel, an energy funnel, down toward the reaction center at that specialized chlorophyll, P680, P700. So that, and it's resonance energy transfer. It's like, you know how tuning forks work? If you take one at a particular frequency and you agitate it and it starts to vibrate, if you have another one at the same frequency, it will excite that because of resonance energy transfer. And this is the same phenomenon of passing energy from one pigment to another in the chlorophyll, or in these, what we call the light harvesting antennae. So they're light harvesting like antennae. But it can't go back because it's downward and a little bit of energy is lost in each transfer down toward the reaction center. When that gets excited by the resonance energy that's brought to it from light excitation, it moves, it donates an electron into a short electron transfer chain. Right? So now you should be seeing these parallels with mitochondrial electron transport. In doing so, it needs an electron back. So those chlorophylls have donated an electron from the reaction center. It has to get one back. The complex there at that reaction center, it uses a metal. So this is just like us. We talked about these metals we need, iron, selenium, zinc, etc. We need them for enzyme activity mostly. So do plants. There's a special metal. It's a manganese. It's there. It's involved in water splitting. Now you're oxidizing water. We looked at reducing oxygen to give you water. Now you're doing the opposite. So there's a parallel, but, well, I guess it isn't a parallel. It's the opposite of parallel. The flip side, splitting water instead of making it. And releasing the oxygen, because you don't need it. Maybe mitochondria nearby, the chloroplast capture, maybe they don't. But now those electrons go back into the system because you just lost some from exciting that specialized chlorophyll. Okay? Those electrons get passed through a series of carriers to another specialized chlorophyll in a different photosystem. Right? It's still all within the chloroplast. They're just different systems. They're mechanistically distinct. But it's the same principle. So an electron is donated to that second photosystem, the specialized chlorophyll, the P700. So why does it need an electron? Because it's donated one to another short electron transfer chain. How did it get excited enough to donate electron? From light. Right? So they don't have a little lightning bolt here. But that's why these are excited, is because these pigment molecules have been excited by light. They excite the resonance energy transfer. They excite that chlorophyll. It donates an electron. Instead of letting the electron come back to its ground state and releasing light and heat, it donates that electron, but it needs one back. It can't split water. So it has to get that electron from that transport chain. So where does that electron end up? Because remember how important that is in mitochondrial electron transport. It ends up in oxygen in the examples we've looked at. Here it doesn't. You're making oxygen. That electron goes to, I put it in this one. I'm going to jump ahead one and then I'll come back. That electron ends up on NADP. It reduces it. NADP is the low energy form of NADPH. You reduce NADP, it gains electrons, it goes to a higher energy state. You end up with NADPH, a high energy compound. But just like, so here's a good parallel, just like the electron transport chain of mitochondria, as the electrons are going from one carrier to the next, that's a downhill, gain, reduction, oxidation, reduction, oxidation. In those oxidation reactions where it loses electron, it goes to a lower energy carrier, you can use some of that energy, wasted as heat, or same as mitochondria, move protons against an electrochemical gradient. But this time it's into the lumen, which means space, of the thylakoid disk. So just imagine a big hollow disk. Build up protons in there, so you build up charge and concentration of hydrogen ions. And now you have a chemical gradient that you can exploit, right? Potential energy, exploit that to recapture it in transformation, and again in potential energy in the form of bonds with the kinetic energy intermediate. So really tight parallel, potential energy gradient, electrochemical, flow, let those protons flow back down through ATP synthase, spin it, capture some of that energy in the endergonic reaction of ADP to ATP, and now you're great. You got two high energy molecules, NADPH, ATP. You can use those to drive endergonic reactions. And because this is the reduced form, this is the reduced form of NADP, it can reduce other compounds. What do you want to reduce? Carbon. Because the carbon you just got is carbon dioxide. It's the most oxidized form of carbon. And it's not good directly as a building block. You have to reduce it to start to make these carbon skeletons that you'll use to build the biological world. So this is just a diagram of that, the manganese containing water-splitting enzyme in photosystem 2. So photosystem 2 is capable of splitting water, no small task, very difficult to do. It produces hydrogen ions that are useful to contribute to the ultimately the development of that potential gradient in the thylakoid disc, and the oxygen can just diffuse away. It's a byproduct, it's not really a function, right? So we'll wrap this up on Friday, but be approaching these with the same kind of mindset. What are they for? What goes in, what comes out? How is it regulated? We'll come to that. And the other one, where do they happen? All right, so that wraps it up today. I'll see you guys on Friday.